Geoff actually did his PhD here at UQ, and he's worked with our with many of the CBCS team in projects related to myrtle rust, especially I know with Martine and the Threatened Species Hub. So he's probably familiar to many people. His role is as a principal forest pathologist and forest production and protection team leader with Queensland's Department of Agriculture and Fisheries. And he has over 20 years of experience in forest health and biosecurity. He's been doing research on myrtle rust since 2010, when the disease was first detected here, and studying the impacts of the disease on both the Myrtaceae as well as associated plant communities. In 2016, Geoff won the Biosecurity Impact Award for making a measurable change to plant biosecurity. And yeah, so with that, I think I'll hand it over to you, and we will look forward to your talk and come back with questions afterward.
Okay, thank you. I'll just get that up and running. Okay, hopefully everyone can see that now. It's all up. Thank you, Matthew. Thank you for the introduction, and thank you everyone for having me come and present today. I'm just going to give you an introduction, I guess, to myrtle rust, a bit of an overview, general overview of what the rust is and the impacts we're seeing here in Australia. First of all, what is myrtle rust? It's a disease caused by the rust fungus, Ostreopuccinia sidii, which was previously known as Puccinia sidii. It's only recently changed over to that new genus, and it's a genus that sits out on its own. It's also commonly known as guava, eucalyptus, or ohia rust, which is based on different host ranges that's affected in different countries. More recently, that ohia rust being one in Hawaii that's affecting one of their iconic Myrtaceae species. It is native to South America, where the impacts have been primarily on either guava grown in plantations or eucalypts grown in plantations as well. Over there, they see limited or no impact in the native ecosystems at all. And that's not uncommon when you get a co-evolved pathosystem. I've got here also the life cycle. It's one unusual in comparison to some other rusts in that it does complete its life cycle on the one host. You can see here from the images on the right there, this is the asexual state, or the Uridinia spores being produced, these beautiful golden coloured spores that you get. Can be quite dramatic depending on your host and how they're responding. In this case, you've got Rhodamnia maydeniana with this reddish coloured reaction, and then the stark contrast with the yellow Uridinia spores. Now the Tilia spores, they're quite dark in colour, can be an orangey brown looking colour. In South America, they suggest that they rarely see the sexual state. Our understanding of the sexual state and its role in terms of recombination and what the pathogen is doing and the impacts in different systems is a little bit unknown at this point in time. There were suggestions that really it was a defunct. It was, while we saw Tilia spores, it wasn't actually causing recombination to occur in the population. But more recent studies in both South Africa and New Zealand suggest that recombination is actually occurring within the populations. We need to now understand, well, how does that maybe influence the host range and the impacts on the different hosts within the different ecosystems? Just looking at the disease globally, as I said, it originated in South America, and we know there are multiple strains, multiple different strains within that site. And we know of at least 10, there's likely to be more. The first big jump of this pathogen outside of its native range was into Hawaii in 2005, followed by 2007 into Japan. That incursion into Japan was actually in a glasshouse system, so it was fairly restricted and didn't actually spread from there. But then the biggest and most concerning jump was obviously in 2010 when it first arrived in Australia. Since then, we've had detections in New Caledonia. There's a unique strain which turned up in South Africa in 2013. And then the most recent one was in New Zealand in 2017. We've also seen it spread up into Asia in 2016. We're not sure of those dates as to whether it was there a little bit earlier. It was actually detected by Australians working over there on biocontrol programs. So it could have been sitting in that environment for a little bit longer than what we've got in terms of that detection date. Now, as I mentioned, there's multiple strains, and if we look at the graph there, it's just demonstrating the spread of these different strains globally. What you'll also notice there, around the 1980s, was when this pathogen really started impacting on plantations in eucalyptus in Brazil, causing significant decline, particularly the establishment stage of plantations and in their nurseries. And that's when it became a major issue in terms of a biosecurity threat to Australia. It was really recognised and put up there as in our top 10 things we wanted to keep out of the country. So there were all sorts of things put in place to limit the chance of rust arriving in Australia, things like preventing Myrtaceae plants coming in without extended periods in quarantine, etc. As you can see from the most recent spreads, the red dots are that of the pandemic strain. That pandemic strain is the strain that we have here in Australia, and it's been responsible for the most recent spreads around the world, apart from that unique strain that's occurring in South Africa. The origins of that unique strain we still haven't detected in South America, but you would assume that it is South America. It just hasn't been picked up in their native ecosystems. Moving on to Australia, it was first detected in April 2010 on the central coast of New South Wales. How it got here is still unknown, whether it was an accidental introduction or through illegal trade, nobody actually knows. But we do know that the initial spread of the disease was through anthropogenic means, through either infected nursery stock or people who are working within these nurseries and going out to help in bush care regeneration programs. There was a clear pattern indicating that was initial spread. So in 2010, December 2010, just before we had the floods here in Brisbane, it turned up again in nursery stock. Similarly in Victoria, again on nursery stock, Central Queensland, North Queensland, all those initial jumps were via detections in nurseries. So movement of that plant material was a huge issue. It's post that initial spread, once it was into the native bushland, natural spread via primarily wind dispersal was the main form of spread and extension of geographic range. It's now well established in native ecosystems along the east coast of New South Wales and Queensland, east of the Great Dividing Range. The conditions for disease development are less suitable once you go west of the Great Dividing Range, once those rainfall levels drop off. In the case of Victoria and Tasmania, it hasn't established outside urban gardens. It's pretty much restricted just to people's backyards, the botanic gardens, that sort of thing. It hasn't got into the native ecosystems. We're not sure whether that's a host thing or it doesn't appear as though it should be a climate. If you look at the conditions that occur in New Zealand and the spread in native ecosystems over there, strongly suggests it's maybe a host-driven thing rather than a climatic thing. In the case of Northern Territory, it is established in native ecosystems there and affecting a range of different species. It was first picked up there in Melville Island in Tiwis. We have had detections on Lord Howe. They detected on a highly susceptible introduced species, Syzygium jambos. They've now removed all that Syzygium jambos from the island, haven't detected rust on there since, which is encouraging. Even more encouraging is that we haven't yet seen rust in Adelaide, in South Australia or Western Australia. And obviously Western Australia is pretty crucial given the diversity of Myrtaceae existing within Western Australia. So host range, Myrtaceae. So any species within Myrtaceae is a potential host for this pathogen. It's a pretty broad host range for a rust fungus. If you compare it to some of these wheat and cereal rust, they're quite specific in terms of the species they'll affect, whereas this affects a whole range of different species. I don't think I need to tell too much about Myrtaceae, but culturally, ecologically, and commercially significant species. They dominate many of our key ecosystems, and I think that's out of 11 out of 13 of the ecosystems in which they are dominant. Commercially, they're obviously very significant. We've been fortunate, I suppose, that there has been limited impact on eucalypt plantation and both native forests. It's been restricted pretty much to nurseries and some indications of minor impact in the establishment phase of plantations. The bigger impacts have been on things like the lemon myrtle industry, where during periods where rust is sort of quite active, we're getting up to 70% production losses occurring in that. It does occur in tea tree plantations, but the impacts are fairly minor. If we look at the host range globally, we're looking at 520 species from 70 different genera. Australia's got more than half those species from 57 different genera. As I said, very, very wide host range that's affected by rust. So the impacts, just generally localised extinction. I'm going to talk a little bit more about native guava. We're seeing native guava disappearing from our environment. We're seeing further decline of species that are already listed as threatened, including the Syzygium hodgkinsonii, where the impacts are primarily on regenerating seedlings and saplings rather than the mature trees. We're now sort of getting indications of changes in plant community structures, and I'm going to talk about this in more detail. More specifically, I guess, in those ecosystems where Myrtaceae are quite dominant. Recently, this thing loves disturbance. So any disturbance factor, whether it be clearing, whether it be cyclone, and more recently, what we've seen with the fire side of things, this thing can take off and build up and spread and impact quite significantly, very rapidly. So what are the symptoms? Huge range. As you can imagine, with all the different host species, you're not going to get a simple, nice little same symptom. It infects only the new growth, the older leaves, the fully expanded leaves, the older shoots, the older stems. Once they're woody, they become totally resistant to infection. We also see infection on fruit, and that can be on mature and immature fruit. We also see infection on all parts of the flower. So the symptoms can range from, and you can see the images here that we've got, from a restricted leaf spot right through to this blighting, which we've got down the bottom there, almost a scorch, as though it's been hit with either a herbicide or scorched by fire. So it's a huge range of things. As I said before, we get different reactions from different hosts. You get this purple spotting. You get some where you get infection primarily on the stems rather than the foliage itself. And we see that particularly with some of our leptospermum species in our coastal heathlands. With repeated infection, we can get significant dieback. We get deformity within the trees. We get this, what we call witch's broom effect, where there's a loss of apical dominant. Rather than being a nice tall tree, these things become shrubs. As I mentioned, infection on all flowering parts, affecting flowering production. So you're getting deformed flowers. We're getting a situation where immature and mature fruits, they're dropping off before they reach maturity if they're infected as mature. We're also getting this odd thing where we're actually getting sporulation internally within the fruit. The picture you can see here is Eugenia rainwartiana, which is the beach cherry, a known food source for the cassowary. We've also got on there a picture of a bee collecting spores. We see both the honey bee and the native bees actively foraging on spores. Now, they are a protein source. We don't understand, I guess, whether they are favouring the collection of spores over pollen, or what effects it might have on high viability if the protein levels are lower than the normal food source. But does also suggest they could be a vector for rust spores from flower to flower. So the environment's affected, as I showed, up and down the Queensland, New South Wales coast. There's a range of different ecosystems in which this thing's impacting. Tropical and subtropical rainforests, we get highland rainforest areas, lowland rainforests and wet sclerophyll ecosystems. You can see the photo on the top left there that Jarrah Wills took. It's showing the decline of Tristania exiliflora, Tristaniopsis exiliflora, sorry. Which is a rheophyte that grows along many of those river and creek banks in North Queensland. We've got the Sand Island ecosystems, World Heritage areas like the Gari or Fraser Island. We've got a range of different species affected over there. And our wetlands and coastal heath systems. There's a range of different species, particularly our Melaleuca quincunurvias, or the paperbarks growing in those wetland areas that are affected. And I said those areas that are affected by disturbance, in particular fire. We've seen significant impacts on a range of different species. So as first talked about that the main impacts of this thing were on seedlings and saplings, but we've actually seen impacts from seedlings through to 100-year-old trees. There's no, we do see some species where they transition out of seedling into mature trees, that they do become more resistant over time. An example is that it seems to be some of the eucalypt species where once they get that mature foliage, they tend to be a little bit more tolerant or resistant to the disease. Starting to look now a little bit more in detail at the impact. We've got some species that are just disappearing as a result of rust. This is a good example, Myrtaceae, which is our native guava. We've seen severe decline across the geographic range of this species. So its range is from just north of Sydney right up along to the Fraser Coast. And before rust arrived, it was considered as common and widespread within ecosystems, and no evidence of any dieback. And yet within 4 years of rust arriving, we saw significant dieback. And we're not talking about sort of these young trees, we're talking about quite mature and tall trees. You can see in the picture there, just west of, that's a site west of Coffs Harbour. Every tree in that site was killed off. And that was, I think, probably 2013, we first observed that level of dieback. Within a lot of these sites, all that's remaining are root suckers. And you can see in the bottom right there where we've got pictures of infection of those little root suckers coming through. So this thing has disappeared from some sites. We have localised extinction where there's just no regeneration whatsoever occurring with this species. We got other species that are in decline. While we haven't seen the same level of deaths within some of these species, we are seeing them gradually disappear or decline from various sites. In this case, this is another common species, Rhodamnia rubescens, one of the malletwood species. Its distribution is similar, although it extends a little bit further south than the native guava, Rhodomyrtaceae. And its impacts, all the populations we've looked at over time are in decline. There are some individuals which show better tolerance than others, but what we do see is that flower and fruit on those particular individuals still get infected. So foliage-wise, they haven't shown that same level of decline, but they're not regenerating. And we've since rust arrived, we've had no evidence of seedling recruitment occurring in any particular site. Again, this was a species that was considered common and widespread, but it's now critically endangered. So significant impacts in less than 10 years. As I talked about a little bit earlier, we're starting to see plant community changes, particularly in sites where Myrtaceae are dominant. This is a site in Tallebudgera Valley. It's a wet sclerophyll site dominated by Eucalyptus grandis and Lophosamon confertus. And it's got a well-established rainforest understory, dominated again by Myrtaceae. 3 of those dominant species are Archaerotomyrtus beckelii, which is a known pioneer species, Decosperma humuli, and Gossia hillii. From the table there, you can actually see the mid-story and the understory species when we first started assessing the site were dominated by Myrtaceae, but you can see that transition across to non-Myrtaceae species. So when we started looking at the regeneration, what was coming through underneath all these dead trees killed off by rust was non-Myrtaceae. That included a lot of weed species as well. So we're getting camphor laurels coming in, we're getting lantana coming, and as well as the ochna arriving into these sites. If you look at the species decline of those 3 dominant Myrtaceae over the years, just the 2 years that we looked at, 2016 to 18, we're getting a dramatic decline in those species. It's hard to find a Gossia hillii or Decosperma humuli present within that site today. Archaerotomyrtus, they're producing these epicormic reshoots, but functionally they're just not performing. They're not reproducing. Recently, we started looking at sort of the numbers of dead trees within these sites, and we've estimated approximately 3,500 dead or dying trees per hectare within this site. It's not. We've then gone and looked as to see whether this site is representative of others. So right through that Tallebudgera Valley, we're seeing similar levels of dieback, and even into areas of northern New South Wales under similar ecosystems. We are getting these similar levels of dieback occurring. So if we start to look at wet sclerophyll, those low-lying rainforest ecosystems, we start to look at the area that covers, that's quite a significant impact that this disease could potentially be having over time. As I said, impacts on fire recovery. As you can imagine, post-fire, you just got perfect conditions for the development of rust. And we've seen impact on seedlings, reshoots, and a whole range of different species. This is a site in Bundjalung National Park in northern New South Wales there. And as you can imagine, this is, I think it's late January. The fire went through in late November. We're starting to get that reshoot. So all that's new flush and perfect conditions for the development and rapid spread of rust. And really it only took till March when we first saw our first infection occurring on some of these species, and just the level of infection was exponential following that. And we're still seeing repeated infection in these sites now, affecting reshoot regeneration and seedling recruitment. We know that rainforests obviously aren't designed for fire, and we saw quite significant impacts on a range of different species within rainforest ecosystems. I've talked about this species, Rhodamnia rubescens, previously, and the fact that it was already under threat from rust before, but what we've seen in those fire-affected areas, any regeneration of the species has been severely impacted by rust. So it seems to be that the combination of fire and rust coming back onto that regeneration is probably accelerating the decline of some of these remaining populations. So we looked at sites from Binnaburra west out to Main Range National Park, into New South Wales in Nightcap National Park, as well as down south into Washpool. And we looked at 96 different trees across those sites, 80% of which had infection occurring on new flush, and 70% have had some level of dieback. Now, we've gone back to these sites since, and those dieback levels have increased. So I haven't finalised the data yet, but we know that the dieback is quite significant, and some trees have been killed off. Melaleuca quinquenervia is another one of those ones that we've been targeting. Both those coastal river systems, inland waterways, and some of these swamp lands where they are dominant and not many other species can replace it if we start losing many of these species. We've got a range of plots set up, ranging from Gari in Queensland down to Bundjalung National Park in New South Wales. So we actually looked at a range of these different sites, looked at the susceptibility of trees based on disease incidence within these sites, and we compared that to the fire intensities. And what was interesting, for this particular species, where we had lower fire intensities or a range of fire intensities within the sites, we were actually getting lower disease incidence and lower levels of impact in relation to dieback caused by the disease. So there's potential, obviously, I guess modifying fire regimes or ensuring frequent burns within these Melaleuca ecosystems may reduce the severity of impact within some of these ecosystems. But we still need to set up longer term monitoring plots to further study that. This just gives you an idea of the decline rates. This is looking at narrow-stemmed paperbark along the Esk River in Bundjalung National Park. So the photo on the left there was taken in March 2020. And then 3 months later, we've pretty much got death of that reshoot. These things will then reshoot, produce another lot of reshoot that then gets in turn infected and results in basically the death of all that reshoot and death of the tree. Just to give you an idea of that progression, so obviously when if we look at the first two trees with susceptible flush, the higher the number of trees with flush, you get perfect conditions, you get a build up in rust populations. You get a decline as rust kills off a lot of that new flush. You get a decrease in the amount of available susceptible flush, so you get a decline in rust populations. Again, you get this up and down nature of infection, but rust just comes in every time it throws that new shoot, so that these things are getting whacked every time they're trying to throw out a new shoot. So what we're seeing now is this progressive increase in the number of reshoot deaths that are occurring on plants. And more recently, we started to see total tree death occurring within these. Interesting, and I haven't got the data in front of me here, but we've started looking at flowering in this recent flowering period, and only those trees that don't have rust on them are actually producing flowers, and that's a pretty small percentage within some of these sites. Melaleuca nodosa, it's a lesser known species, it's quite common across some of these sites. It's in terms of its ecology, there's not that much information out there, but it is quite dominant in some of these heathlands, and it does also grow as a small tree once you go into some of these more inland sites. Post-fire, all sites that we looked at were affected, and quite significant levels of dieback following the repeated infection of the new shoots. Hard to photograph. You can see those two photos on the far right. I just took those last week. So with the repeated infection of these new growing tips, these things are becoming suppressed within the ecosystem. Non-Myrtaceae are taking over, and so it's hard to actually find some of these things. So they just can't compete with these things. Are not subject to rust infection. One of the more concerning things we've seen is that the level of tolerance within the populations is quite low. We're sort of looking at less than 30% of individuals that seem quite tolerant to the disease. But also the impact on the flowering. So those that have got high levels of dieback, we're seeing significant levels of reduction in flowering. And so we've done this based on just the percentage of branches that are producing flowers. So you can see, particularly on the images there, top one showing one that's more tolerant to disease, which has got very little level of dieback, significant impact on significant levels of flowering occurring on all branches, compared to that one below where we've got dieback and we've got only scatterings of flowering. So these things, a lot of the melaleucas obviously rely on generalists for pollination. So if we start interfering, I guess, with some of that process, with a decreased frequency in flowering between individuals, does that impact on the pollination process. But also, we're looking at sort of a quite considerable narrowing of genetic pool if we've got less than 30% of individuals showing tolerance to the disease. So there's a lot of work we need to do to better understand what the impacts are for species like these. I put this in there. One of the things that I guess we don't have a good grasp on is the impact on seedling recruitment in general. We had looked at some of the impact post-fire on seedling recruitment, but again, it was difficult. With the COVID restrictions particularly, we could only travel sort of every month rather than sort of regularly. So it was hard then to track the levels of impact within seedling populations. Even some of the individuals we did see, on the photo on the left there is Melaleuca quinquenervia. We did see a lot of seedling regeneration coming up, and some actually reaching that sort of hip height and looking okay. And then more recently, when rust levels have really increased, these things have got smashed. And you can see there the apical dominance has been lost, it's becoming shrubby. We don't know whether this tree will survive, whether it'll just continue on being a change in different form, tree form, whether it'll get killed off as a result of that repeated infection. We just don't have the data on that. On the far right there is Eucalyptus pileolaris, or the blackbutt. It's the first time we've really ever seen heavy levels of infection on eucalypts was post-fire. And we saw within the seedling populations, probably it varied from site to site, but we're looking around 20 to 30% of individuals showing some level of infection. And we're probably looking at a lower number, maybe 10% that were actually killed off by rust during the study period. Again, some individuals disappeared, but we couldn't ascertain whether that was rust or whether there was a combination of things occurring. Most recently, and this was only 2 weeks ago, we were on Gari working with the Butler Land and Sea Rangers. And it's the first time we've ever seen infection on Syncarpia hilii in native ecosystems. Now we're looking, again, pretty low levels of infection at the time, but because it was just a snapshot in time, we did see individuals starting to get infected, but at only the early stages. So we just don't know what the levels of impact will be within that population. So it's something that we're hoping to follow through. But culturally, hugely significant species, but obviously ecologically a hugely significant species that actually relies on disturbance for regeneration. So that's one that we're going to have to keep an eye on. And I was talking to a few people earlier and saying, every time we go out in the field, we find something new, and this is just an example. I've been on Gari doing surveys since probably 2013, never seen any infection on this particular species, and yet we saw quite a bit on this trip. So just to wrap up, and just so we're not leaving it all on a sad and depressing note, there is good news. As I mentioned earlier, Western Australia and South Australia are free of myrtle rust, and there's a lot of plans in place to maintain that. There's strict biosecurity processes being put in place in Western Australia, and then survey processes being implemented so that if it does turn up anywhere, they can actually try and eradicate it before it establishes. There is resistance at a species level. We know things like the brush box are quite resistant to disease. We've never seen any infection. We can't get it to infect in the glasshouse. Within species, we know there's quite a good level of resistance, particularly within the Eucalyptus and Corymbia, where because of obviously the interest from a commercial perspective, there's well-structured populations in collections. And down to a family level, we've identified resistance within all species. What's interesting with a lot of the eucalypts and the corymbias is that the inland provenances appear more resistant than the coastal provenances to rust, whereas a lot of the endemic pathogens, it's the vice versa. So adds a layer of complication. In Melaleuca quinquenervia, we've actually done all the broadleaf Melaleuca species, but Melaleuca quinquenervia, there's a range of different susceptibilities within different populations around New South Wales and Queensland. It ranges from anything only 30%, 20 to 30% resistance, right up to 80 to 90% resistance within populations. And there's a little bit of work being done by other students looking at sort of what factors might be associated with resistance within individuals and populations as well. There are germplasm collections both here in Queensland and in New South Wales, but it's primarily based on those 3 species at this point in time. One of the big complicating factors is that a lot of these are obviously fleshy fruited species. So how do you conserve a large number of different species or even a large number of individuals of a single species? There's not a structured population collection where sort of there's a few targeted ones at this point in time, but we're trying to encourage different land care groups, different Indigenous groups to start collecting, I guess, learning lessons from myrtle rust in terms of what might happen if we get other pests and diseases arriving from overseas and the threats that might pose. So collecting seed of some of their more common and dominant and ecologically important species and maintaining those for regeneration programs. National and international collaboration. So it's a key in terms of identifying where these other rust strains might be present so that we can actually identify pathways. If we get one of those other strains arriving, the risk is that they might have a new host range. We know in Brazil the indications are every 15 to 19 years they get the emergence of a different strain that overcomes a lot of their eucalypt resistance that they've built in within their populations. We don't want that here. The other risk is that if we get another strain coming in and we do get recombination occurring between the strain we have here and another strain, that might change things as well. The biggest problem about waiting until something gets here before we worry about, we're never going to be able to identify it to be able to eradicate it once it's in the country. The symptoms are going to be the same and there's no way you can establish a eradication program based on that. So really we need to shut down the pathways. We know post arrival of myrtle rust, they restarted the introduction or the importation of timber from Brazil, including eucalyptus timber. And that in 2006 was identified as a high-risk pathway with live spores having arrived on both the timber packaging and containers associated with that. So we're working at the moment to try and review that, but I think it is key. The other key thing about those pathways is not just us, but all the other countries around us must look at sort of the pathways, the risk pathways, because if it gets into our region, it can easily distribute via wind dispersal. We are working with other countries on rapid diagnostics. At the moment, we have no way of determining whether we have other strains here in Australia. We just don't have the genetic material to be able to do that, but there is work going on in the US to try and develop LAMP assays to assist with that. We do need to know if other strains are here because it does then influence how we might look at sort of resistance screening for populations, whether that be from a commercial species or whether from the ecological, we're looking at regeneration of species. The biggest problem we have in terms of, I guess, looking at this and how we manage it is we don't really have a good understanding of the impacts from a broader ecological perspective. I mean, we've looked at impacts in terms of individual tree species, but we haven't looked at that broader impact. What are the flow-on effects in terms of the way we're doing, how we, I guess, rating impacts of the loss of a species or the change in an ecosystem? We just don't understand that. We haven't looked at the flow-on effects, so losing all these species, what was reliant on that, and from a pollinator's perspective, something that feeds on those. In that site in Talabudra, we would have had a massive understory and mid-story of flowering and fruiting individuals. How has that changed the process in terms of those flow-on effects for pollinators and those insects, birds, flying foxes that are relying on those for food sources? It's something we need to look further into. The other thing is with the Melaleuca quinquenervi, if we're reducing the level of flowering within populations, what sort of influence does that have on that species as a food source? Very quick thank you. There's a number of people I've worked with over the years. We started off as a very small team. Angus Carnegie, Fiona Giblin and myself pretty much running around with Gordon Guymer doing IDs on Myrtaceae and trying to work out the host range of this. But there has been momentum more recently, and it's been nice to have a whole team of different people working both here, and we've worked closely with guys in New Zealand. And more recently, we've started working with different Indigenous groups, both on Gari and down around the northern New South Wales area. Particularly like to thank the PBCRC and Plant Biosecurity Science Foundation as well, and the ANPC Threatened Species Hub and DOR for their contribution in terms of funding. Thank you.